So, thank you for accepting me and inviting me to this talk for the Rust scientific community. I would like to present myself as Gaurav Sablok, and I am working as a bioinformatic area expert at the Institute of Bioorganism Policy Academy, NOIC. So, it is an institute of bioorganic chemistry, and it has been ranked at A plus institute in Poland. So, I work here providing the genomic analysis for all of the services for mainly for now for the human genomics, and in the past I have been working on the plant genomics, metagenomics, metatranscriptomics, and the from Illumina to long read sequencing, genome assembly, and genome annotation. So what I'm going to present today is a Rust application. The idea for Rust application was conceived by Professor Louisa Hansuch, and I am mainly responsible for the development of this application. So I am going to present this now. So if I go to the next slide, and this, so the key features why I choose the Rust for this, I have been a principal coder in previously in C++ for many years, and after that Python, and now Rust and TypeScript I use mainly, and with R for the data analysis. So why I choose the Rust? Because it is a memory safe, it has, and the application is available for Conda. It is an asynchronous SQLite application. What this provides much scalable analysis. So eVai is a variant annotation platform that provides the annotation of the VCF files using several previously published databases. But the state of the files is the analysis files, which is provides, but after the analysis file, it is very difficult to go if you want to do a population scale analysis, or you want to implement some stats feature, or you want to get variant summaries or annotate variants with the sequence information. So the my group lead and the laboratory lead, Louisa Hansel, they conceived the idea, and I was the principal responsible for the coding of this application. So what it does, it provides you a scalable application for providing the population scale analysis. So the features that have been implemented in this, if you go to the next slide, thank you, Matthew, for this. The features that have been implemented in this, it basically comes with two state of point application. One is the eVaiutilities, and second is the VCF filter file. So the eVai produces two kind of files. One is the eVai utility showing the ACMG configuration, ACMG standards of annotating the variants with several of the features such as the alternate and the reference alleles, transcripts, and pathogeneticity scores, databases, and how much of the score is the coverage, the quality, geno quality, and several other features which are embedded in the BCF filter, which are embedded. But on the front page of the eVai, you can search them across using the web. You can search them across one sample. You cannot search them across a multi-scalable. So you need something which can be scaled to millions of samples at one time, which can be scaled to thousands of samples at one time. So here the eVai utilities comes handy in. What it provides several of the various features that are enlisted in the right side of the slide, such as the variant report generation. You can filter according to the pathogeneity score, variant filter. You can create the specific database for the patient system management. You can analyze the GTF, provide the variant sequence annotation. Plus, it supports the version of the eVai, the old version also, as well as the new version also. It gives you the ACMG transcript and also the filtering according to alternate and the reference search. I will, in the next slide, I'm going to go through the some of the features of the VCF filters and eVai utilities to illustrate as what it shows, and how we can use to illustrate, to get the information that we are pondering upon, and we can make the way out of it. So if you go to the next slide, thank you, Matthew, for this one. So the BCF filter is on the left side of the slide, and the eVai analyzer or the eVai utility is on the right side of the slide. So if you go to the VCF filter, in the VCF filter, there is a default VCF filter option which filters the VCF file according to the multi-zygosity with a quality value of the 40, and the variant search of a with the allele search. And in the bottom down options, you have more options, such as the how to filter the variant according to the VCF, along with the variant and the quality, along with the variant and the coverage, along with the covariant and the zygosity. So it means you can select a particular point of a variant, you can select a particular zygosity, and you will get a reports only for that particular variant and with that zygosity. So what it does? You can provide in all of these options, you need to provide one folder which has all of the VCF files for one particular clinical study. So it will go through all of those, whether that folder has 1,000 files, millions files, for or all of the files involved in one particular clinical study. It will go through and will give you information spanning across all those files. So going through each and every file in the eVai as a web interface is not possible, and it will give you a haggard look, make yourself haggard. But with this you can go through very conveniently, spontaneously, and you can get a reflective view of what you want to do with it. So when you are going, you can provide the particular folder, you can give the variant, and you can give the coverage or the zygosity or the quality, and it will produce the filtered variant according to that particular file. It will produce the filtered files as well as the file before, prior to filtering, and after filtering the stats on it. It will give you mean, median, and the standard deviations of the presence of the variant across those particular samples. So you get, plus it will also give you how many of the variants are unique variants, and how many of the and how much of the reported presence of those particular variants across each of those particular files, which can be easily plotted into the R. The right side of the version which shows the eVaiutilities, that is a much pronounced enhanced version which uses the ACMG files. So there also, each of the options, you just have to provide the folder for each of the particular sequence in particular clinical study, and then you can provide the options and with the particular folder, and you can search for the population specific variant across the population. You can search for way that give me these variants within the code, within these coordinates. So you can search with the option coordinate search, that will give you variants across all those file within these coordinates. And then if you say I want some particular pathogenicity score or the annotation, then you can search for a particular pathogenicity score and get those variants across all those files. Plus you can search according to the alternate allele, reference allele, and a combination of both alternate and reference allele across all those files. So it makes your work easier when you do a population scale study, to understand how these all of these functions are using the Rust asynchronous. And now I am working on a second version which is going to use a Svelte to give you a web interface for this, so that you can have a clickable and editable variants in case you find information which you can update it. So that version uses Svelte and Rust to give you an updated version. Next slide. And that's what, thank you for, I think that was the last slide. Yes. So thank you for giving me a chance to present the eVaiutilities and the VCA filters. So the main goal of the eVaiutilities and VCA filter is to handle the post eVai files analysis, and to provide you a much robust combination of what the paper has been submitted to the JOS, and it is currently under waitlisted, and I hope to get it quickly under review. And the VCF filter and the Svelte repository that we are planning to publish soon, because it is going to be a Svelte plus Rust web application in which you get the all and it is a desktop application in which you get the all variants annotated and filtered through your desktop clicks. So the people who don't like command line, they can have a web interface, plus it will give you a ready to use display graphs along with the information and annotated charts about the variants. Thank you so much.
Thank you very much for your talk. I do have a question. What was the main challenge in terms of coding for eVai?
The main challenge was, basically, I tell you, means I have been writing in uh, the type languages since the start of my doing work. So when I started writing Rust last year. So prior to that, only C++. So for me, the coding challenge, I really don't felt any kind of a particular coding challenge in this, means coding difficulty in this, or understanding or what I need to implement. Yes, for me, difficult was how to understand the biological interpretation of this, and how to make sense of the data. So I had a rapid communication with the people I presented here, several meetings. I got very valuable and feedback points to added, and that allowed me to actually conceive how to develop the state of the art softwares for human genomics, because previously I have been doing it for the plants and metagenomes. So the now with the asynchronous application, now I'm implementing. I have previously used Rayon. Rayon is good in implications of this, but the problem with the Rayon is it still lacks some of the features. For example, byte size manipulation, you can it is very difficult to do in Rayon, because it has only two modules, two only applications. One is creating the parallel iterator, one is creating the par iterator, and one is creating the parallel bridge. So when you are doing a parallel iteration, par iteration, or the parallel bridge, you cannot smoothly go through a millions of file at once. So now with that limitation in the Rayon, I have shifted to the using the Tokyo async. And with the Tokyo, but the problem with the Tokyo async is that when you implement a Tokyo async on a main function, it gives you an external route that the parameters of this can be safely sent through threads. So when you're using the Tokyo async, you have to use the clap, which I'm using for the command line building, in a different way that needs to be done. So these are the limitations that I have overcome while developing this, shifting from Rayon to Tokyo and using async standard as the default ones.